All right. So today I wanted to talk about stem loop structures and their role in mutation. So stem loop structures occur in mRNA. Okay, so mRNA is single-stranded, right? So unlike DNA, DNA is double-stranded. It's base paired, and that keeps the code stable, keeps it from incurring mutations along the way. But RNA is a little bit more unstable. It's also got um, um, so it makes when it's single stranded it has it's more dynamic, and in fact DNA occasionally when it's being transcribed or translated while it's in a single stranded mode, it also can do the same things. Okay. So this structure is what we're looking at here is an RNA one, but the same thing happens in DNA when it is single-stranded. Okay, so this is a stem loop structure, and so basically in a stem loop structure, this happens when you have going one way, you have a set of base pairs, and as you follow the sequence, you run into the complementary base pairs going the other way. Okay, so you can see that G lines up with C, C lines up with G. So this. We can think about this when we think about this from a DNA translation standpoint. This is all going in a single line, but when it actually gets into 3 dimensions, um, these things actually naturally bond to each other. Okay, and so the thing about in the middle of this loop, you can tell this is RNA by the way because it's got uracil instead of thymine. Um, but the interesting thing about this is that this part right here that's bound to itself is very stable. This part where you have unpaired bases, since these are not paired, they're actually a lot more mutable. And so that's true of both RNA and DNA. And so one thing that Barbara Wright pointed out, let's see here. So it started with this paper, which is very interesting. It's a biochemical mechanism for non-random mutations in evolution. It was published in 2000. And so in this paper, she points out that this might be a mechanism by which mutations are directed at certain sites. So when DNA gets single-stranded, when it's being copied, or when it's being copied, or when it's being translated, uh, it itself forms these stem loop structures, which then has regions of the DNA which are not likely to be mutated because uh, they're folded over on themselves and are in a stable configuration, and then base pairs which are much more likely to be mutated. And so basically, this allows the cell to kind of point out, say, hey, look, here are some mutable base pairs. Here's some things that are where mutations can be beneficial. And Barbara Wright has actually done a lot of research on these. So if you look up Barbara Wright and mutation, you can actually find a lot of interesting information, and a lot of this stems from these stem loop structures. So she's done a lot of work on that. So that's one mutational mechanism. I think it's really worth looking at. And so basically, again, it allows the DNA sequence itself to point to where um, likely locations of mutation are. And I think she actually created software to identify these. Um, and Anyway, the other thing to recognize is it's actually the degeneracy of the genetic code that allows for these stem loop structures to exist. So so you can think about it's interesting to think about how the fact that multiple uh, multiple different uh, base triplets code for the same amino acid allows for some freedom in where these stem loop structures are made. And so, anyway, um, I found all that interesting, and it's one of the mechanisms that's used. Uh, I think this is one of the mechanisms that's used by DNA to point to where the mutable sites are. So anyway, that's all for this talk, and I will see you for